The purpose of this video is to illustrate SPSS factorial ANOVA using the TeenSurvey data. Our dependent variable in this case is going to be vocabulary score, and we are going to be using gender and socioeconomic status as our independent variables. In this case, we're going to see whether or not vocabulary scores are stable for both genders. That is, our null hypothesis is that the gender for males is equal to the gender for females, and also that the mean for vocabulary score will be the same for low, middle, and high SES students. And finally, because it's a factorial ANOVA, one of the important hypotheses that we will be checking is that there is no interaction between gender and socioeconomic status in terms of vocabulary score. Okay, so we're going to be testing three null hypotheses: a main effect for gender, a main effect for SES, and the interaction between gender and SES. Gender has 2 levels, SES has 3 levels. To better understand what those levels mean, we can go over here. We see that over to our variable view, and we can see that males are coded 0, females are coded 1. And we can also go down to socioeconomic status, and we can see that low SES is coded 1, middle SES is coded 2, and high SES is coded 3. The next thing that we do is we go to Analyze, over to General Linear Model, and to Univariate. These were some analyses left over. So in this case, our dependent variable is going to be vocabulary, and our independent fixed factors are going to be gender and socioeconomic status. Okay. After we establish our dependent and our independent fixed factors, we're going to make sure that the model that we have is established. We want to make sure that it says full factorial. We want to make sure that the sums of squares are set to type 3. One of the things we want to make sure that we are able to do is to understand what any interaction that occurs looks like. So we go to plots, and we're going to run separate lines for gender, and we want SES along our horizontal axis. The purpose of that will make sense when we start looking at the actual plot. Click on Add, and then Continue. We also want to consider any post-hoc tests for SES that may occur. If there is a significant difference for SES, we want to track down where those significant differences occur. We don't need to do that for gender, since gender only has 2 levels. Significant difference that emerges will be clear enough in terms of the nature of those means. So we click on post-hoc, and we want a post-hoc test for SES, since there are 3 levels of SES. We're going to run the Scheffé test on SES, and we will click Continue. In addition, we go down to Options, and we want to make sure that overall relationships are there, so we can get the means for the gender, means for SES, and means for gender by SES. We make sure that descriptive statistics and homogeneity of variance tests are both checked. We click Continue, and at this point we're ready to run the results. What we see is we have 155 males and 145 females. We have 58 individuals of low SES, 141 of middle, and 101 of high socioeconomic status. We look at these means within gender broken down by socioeconomic status. So in terms of males with low SES, their mean vocabulary score was 46.4. For those males of middle SES, their average vocabulary score was 51.14, and for those of high SES, it was 58.7. We look at the same relationships that are occurring down here with female. We can see just visually that there are differences as we move from low to middle to high SES just based on looking at these means. We can also look down here to see across gender that relationship holds. The next thing that we do is we consider whether we have met the homogeneity of variance assumption. This homogeneity of variance assumption is what allows us to basically pool our variances across all of the different cells in this study. Since our significance value is larger than 0.05 or 0.01, we fail to reject the assumption of homogeneity of variance. The null hypothesis is that the variances are equal across groups, and we fail to reject that assumption based on this. So as we go down here to our Anova table, what we're looking at here is we have gender. This is the main effect for gender. This is the main effect for SES. Below that is the interaction term between gender and SES. Error is our within cell variance. And we have our total, sums of squares total right here with our n-1 degrees of freedom. We have 300 subjects, so we have 299 total degrees of freedom. 294 of those degrees of freedom are associated with within cell variability. We have 1 associated with gender. Since we have 2 levels of gender, we have 1 degree of freedom. We have 3 levels of SES, so we have 2 degrees of freedom for SES. When we multiply gender degrees of freedom by SES degrees of freedom, we get 2 times 1, or 2. We look at these results, and what we see here is we have a population variance estimate for differences between the means associated with gender and the grand mean of 16.456 squared units. For SES, we have 3,038.4 squared units of variance, population variance attributed to differences between socioeconomic status and the grand mean. We have 47.762 squared units related to the interaction, and our within cell variance is 78.484 in terms of our population variance estimate. To get our F calculated scores, what we are doing is we are dividing mean square or population variance estimates for gender, SES, and interaction, all by the within cell population variance estimate. So we divide 16.456 by 78.484, and we get 0.21. We divide 3,038.4 by 78, and we get 38.714. For the interaction term, we divide 47.762 by 78.484, and we get 0.609. These are our F calculated values. From these F calculated values, we can determine the significance values. These are the probabilities. So in this case, a difference of this magnitude that was observed between gender means and the grand mean would have occurred due to random sampling fluctuation alone about 65% of the time, whereas the differences between the means by socioeconomic status and the grand mean, those differences would have occurred due to random sampling fluctuation alone less than 1 time in 1,000. The interaction term, in this case, we have no evidence that there is a significant interaction. If we used an alpha of 0.01 in this case, since our sample sizes are fairly large, we would fail to reject the null hypothesis for gender, we would fail to reject the null hypothesis for an interaction effect, and we would find that we would reject the null hypothesis for socioeconomic status, which indicates that we believe that the means for socioeconomic status are significantly different. We can go down to our socioeconomic, our post-hoc analysis to determine where those differences are, and it appears that we have differences between all groups. That is, between low and medium, between low and high, and between medium and high. There are significant differences in all comparisons. The Scheffé breakdown this way confirms that we have three separate homogeneous groups. When we look at the profile plot, what we see here is while these things appear to intersect with each other, the challenge that we have is that they're basically overlapping with one another. That is, the test for interaction is essentially a test of parallelism of these profile plots. So what we're learning is that because the difference was not statistically significant, it's essentially arguing that these lines are essentially parallel. In this case, we don't really have any evidence to suggest that these lines are different, but the interaction test is really a test of parallelism. And that's basically the rundown of how to interpret a factorial ANOVA.